You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and the Future Tech Health podcast. I have Kunwoo Lee, the CEO and co-founder of GenEdit. And GenEdit deals with gene therapies. They're working on the next generation of non-viral gene editing-based therapeutics. So, Kunwoo, thanks for coming. How are you doing?
Hi, Richard. I'm so glad to be here. Thanks for inviting me.
So tell me, what was your motivation in creating GenEdit? What's a little bit of the history of your work and yourself?
The genesis of it was all because of the very interesting genetic engineering technology. Kind of like, if we think about human being, we are born with DNA sequences that we inherit from our parents. And there are some mutation happen when you are born, but still, after you are born, it is very extremely hard to actually change DNA sequences that you are already born with. And especially if you're already born with genetic mutation that causes some kind of genetic disorders. Previously, there were not that many possible options that you had, but now the new technology, for the gene therapy and gene editing, is actually changing how we cure many types of rare genetic disorders. So that was a very big motivation for us to start this company, GenEdit.
So are there any particular disorders that you're targeting to fix, or is it more the mechanism of how to alter people's genes itself is that you're working on? What's your focus?
There are multiple rare genetic disorders, and we have studied Duchenne muscular dystrophy, Fragile X syndrome, and ARPA1 antitrypsin deficiency. And those are the ones that we had a great success applying the gene editing technology. But what I want to emphasize is this is really a tip of iceberg. And using a gene therapy and gene editing approaches, there are hundreds of genetic disorders, and also some of the disorders beyond genetic. So as we are seeing a lot of cell therapies nowadays, and all those are the potential application and also areas that gene editing and gene therapy can be used. And to accomplish that type of goal, one really missing piece is a delivery technology. And our company is specialized for the delivery of the gene therapy and gene editing molecules.
And just to clarify, are you attempting to modify, babies in utero before they're born? Or is this, can you work with adults potentially, they would inject or take some kind of therapy and it would change some or all of their cells' genetic code in a certain area?
Yeah, that is really great question, because that is something that also public is quite cautious about where the technology is going. And obviously, for most of the pharmaceutical companies, including GenEdit, we focuses on treating adult patients or even juvenile, or adult or juvenile. I may just say one more time here. So kind of like, just like most of the pharmaceutical companies in US, GenEdit also focuses on treating patients who are already born with rare genetic disorders. I mean, there was an attempt in China that even kind of considered editing embryo, but still like kind of going into that early application, there are a number of ethical issues and technology hurdles that we really need to overcome. But kind of we focus on treating patients who are already born with it.
Well, this seems like a really difficult thing because let's say a particular disorder just affects your heart, and you go in there to edit the genes that the heart cells have. Okay, that's one task. If it fails or it goes wrong, it could kill the person, which is obviously terrible. But if you need to modify every cell in the body, and there's like 200 plus cell types, how would you do that? How would that cascade of change look? Would you start with certain cells and then over time it would do more and more cells? Like, how would we ever change the genes of every cell type in our body? And is that needed for most disorders, or are disorders confined to like one organ or one system?
That is a really important question. And currently, I would say that it really depends on disorder that we are talking about. Let's say that if we are talking about hemophilia, we just want to change the genetic sequence or provide a new gene to liver hepatocytes, because those are the cells that produce the factors which are required to cure the disorder. And also, like, there are many like disorder ataxias, or like let's say the Fragile X syndrome, and those are the one that is very important for the CNS, central nervous system. So we want to deliver to like nervous system, but other parts of the body we don't really need to change all the other genes. And the question like kind of becomes, how do we deliver to the target tissue? There are many disorders, and there's like some affect heart, some affect liver, some affect blood cells, and some affect nervous system. And so that we want to develop a technology that actually deliver a gene therapy or gene editing to the target tissue. That has been a huge challenge in the field for the last 20 years. And then there are a few solutions that people have using virus systems, and also like GenEdit's perspective, we are focusing on non-virus systems to address that delivery issue.
Yeah, I guess one way is that, right, viruses can infect cells and inject their DNA into them, and that DNA can get incorporated sometimes into cells. I've heard that we have, endogenous retroviruses in our DNA sequences, right, that we've had for
that is right.
Maybe millions of years. So, okay, so instead of virus vectors, you want to focus on what, bacterial vectors like CRISPR-Cas9?
We are focusing on non-virus systems. So like the backbone of the system, we call it polymer nanoparticle system. So maybe like I can make a simple comparison. As you mentioned that like some of the retroviruses already integrated their gene in human genome, and it happened millions of years ago. And those type of the integrating viruses, the challenge that we have is that we don't know where it gets in. Let's say that if it goes into the sequence that can potentially cause cancer, it is going to be a huge problem. Because of that reason, we want a system that we can have a precise control on where we potentially change the genetic sequences or where we deliver our genetic components. And our focus is a non-virus system. Virus has been and evolved to like transfect people, but still our immune system has developed against those viruses. And some of the viruses like do not really get along with human body when they are injected into the like kind of blood. And so that like what we want is like synthetic delivery system and that encapsulate the cargo quite nicely and then deliver to the target organ of our interest. And then in like many cases, we have delivered nucleic acid that encodes quite important gene, which is missing in some of the patients with rare genetic disorders. And also some of the other cases, we have delivered CRISPR-Cas9 and other types of genetic seizures to precisely change the sequence that has a mutation for like that many patients carry.
Okay. So you were starting to describe the mechanism by which you're going to affect, the cells. You said it was some tiny particles. Can you rename it and talk about that mechanism a little bit?
Yeah, sure. So the approach that we are taking is using a polymer nanoparticle system. And kind of we name it polymer because like it has many different structure, and the rationale is simple. If you think about the antibody is composed of all different structure of amino acids, and but by having millions of the combination, antibody can target almost any types of the cell. And what we do here in GenEdit is that we make thousands of the polymer nanoparticle system, and we test them in the primary cell that is like that is from the organ that we are interested in. And also by doing in vivo testing, we screen out the polymer that interacts with the organ that we are interested in. And also, like scientifically, there has been many studies testing a particular small molecule or peptide sequences that has demonstrated that it can bind to specific cell type. And then we incorporate those type of the structure to our polymer nanoparticle so that we can deliver our cargo to the target tissue.
What are you trying to deliver? So you're trying to bind a substance to a polymer nanoparticle. And I guess your first goal is for the nanoparticle to navigate the cell membrane and be taken inside the cytoplasm. Is that what the mechanism is for a start?
That is right. And there are a number of mechanisms like that nanoparticles rely on. One part is like receptor-mediated endocytosis. If a part of the polymer interacts with the receptor, which is presented on the cellular membrane, then the cell like induce endocytosis to take off the nanoparticle. That is one part, and there are also like many different mechanisms called like pinocytosis and a number of others that also like take off the nanoparticle. So relying on those mechanisms, like we deliver the nanoparticles and the cargo inside to the cell.
So endocytosis, what does that look like? What does a cell do when it utilizes endocytosis? Does it, open up a part of the membrane, or does it give off some kind of fluid-filled vesicle that engulfs material and pulls it inside? What does it look like?
Yeah, that's what you described is like quite accurate. So the things that happen is like just like project a part of the cellular membrane and then take it off and close. And by doing that, it forms a vesicle called endosome, and the nanoparticles goes inside that specific vesicle, and then like cell try to like digest the things inside the endosome. And one feature of our polymer nanoparticle system is that in that endosomal environment, the polymer interacts with the membrane to release the cargo into into the cytoplasm, so kind of releasing the cargo outside endosome.
Okay, so this package is taken inside the cell, and then what happens? Is the cell somehow inspecting the contents of it? Is it segregating it in the cytoplasm? Does it move towards the nucleus? Like how would this package act once it's inside the cell?
Yeah, so inside the endosome, like it starts changing the pH. So like it becomes like more and more acidic condition that helps like digest of the molecule. And then polymer in like a bit acidic condition, it start interacting with the endosomal membrane. And that lipid structure becomes loosened by the polymer interaction. And then kind of things which was inside the endosome is released to the cytoplasm, so that in our case, we deliver kind of many different types of the nucleic acid. And let's say, like, for example, like if we deliver mRNA, the mRNA is like delivered into the like cytoplasm, and that is like where all the translation happens, so that all the like using the kind of translational machinery inside the cell, the mRNA produces the gene that we are interested in. Let's say there can be some functional protein that can do the function that is missing in rare genetic disorders.
Okay. So mRNA is essentially the goal to get inside the cell, and the mRNA will interact with the DNA, and will it rearrange some of the codons? Will it delete some of the codons, snip them out, or what will it do?
So yeah, like your question, maybe I can clarify a little bit. So let's say that
Sure.
Yeah, there are like two different approaches. So one approach is a gene therapy. Gene therapy is a like an approach providing a gene that is missing in the patient. And then in this case, like producing the target protein is the goal. And then that is a way of treatment. And the second case is a gene editing. And gene editing approach, what people do is providing a genetic seizures. And using a genetic seizures, we target a very specific DNA sequences. And recently, many people use CRISPR-Cas9 for this purpose. And we cut the sequence with CRISPR-Cas9, and then we change the specific DNA sequence so that now the cell has a DNA that is repaired, so that it can produce the right protein that we want to cure, so that it can be used to cure genetic disorders.
Interesting. So how are you testing this? Are you using mouse models, or what's the
Yes.
setup in the lab to test this?
Yeah. So like kind of currently there are a number of different like kind of studies are going, and we have kind of our first paper was a CRISPR Gold paper, which we published in Nature Biomedical Engineering 2017. And in that paper, we described that Duchenne muscular dystrophy carrying mouse can have a genetic editing as we deliver CRISPR-Cas9 and the donor DNA sequence to it. And that was a type of the mouse study. And also the other part of the mouse study that we have published was a kind of Fragile X syndrome mouse model, which was like conducted together with our collaborator. And in that study, we used a mouse model that has a mutation causing a Fragile X syndrome. Fragile X syndrome is a kind of big part of the autism, but we clearly know a genetic cause, and a part of the chromosome is like condensed because of a specific mutation. So what we have done was like we have shown that CRISPR-Cas9 delivery into the brain and can actually change a kind of genetic sequences, and that can actually change the behavior of the of the animal. In this case, Fragile X mouse model.
So how long after you administer the therapy do you see changes in the mouse?
So the genetic changes after the injection, we start seeing from next day. And usually all genetic modification is completed within a few days. And then we start seeing the mRNA changes and the protein changes following. And once those kind of effects establish and cells are good and start doing all the right thing that we expect, that is the time that we see actually functional changes of the system. And in those studies, we have followed the mouse for a few months and then check that we continuously see the effects.
Oh, so once the effects happen, they seem to be permanent?
So that is a really important question. So either one injection can be a permanent cure. So that depends on the organ or cell types that we are targeting. So let's say that if we target stem cells and those stem cells linger inside our body for really long periods of time, so that we are gonna have kind of therapeutic effect almost permanently, which is fantastic thing. But currently the technology is still evolving, and we need to do really long-term studies what type of the cells that we are targeting. And in our case, let's say kind of muscle injection, and muscle there is some level of regeneration of the muscle, so that if we are only targeting muscle myocytes, we may have an effect until the muscle regenerates, which can be months to years. But if we targeted muscle stem cell, the effect can be almost permanent. And the other case for the nerve system, the neurons are the one, there is no regeneration, or almost no regeneration. There are very few population of neural stem cells, but it is in very limited area of the brain. So in those cases, the effect of gene editing can be assumed to be almost permanent.
What about tissues that have fast dividing cells versus very slow dividing cells? How do you expect this would have an effect in a tissue where, new cells are created every day versus ones that take, that will last for years?
Yes, I think that is a very interesting question, because that is kind of biology that we are facing. Let's say that hepatocytes are the one that divide very quickly if there is any damage into the liver. And in those type of kind of fast dividing cells, kind of delivery of the kind of Cas9 is easy, and also the kind of gene editing, cutting the sequence with the DNA with a Cas9 is a lot easier. So, and that is one part that people are seeing differences between a non-dividing cell to the fast dividing cell, and biology is quite different. And that is something we need to do further study to fully understand. But so far, many of the scientists who has worked with CRISPR-Cas9 at least show that cutting the DNA sequence has been quite efficient in most of the cell types, regardless non-dividing and dividing. But obviously, there is a difference in terms of efficiency.
Okay. So this is appearing to work in the mouse models. What would be your next steps? How do you get this into, do you want to test it on more different types of animals, or how do you move
Yeah,
forward, let's say, with a clinical trial to test this in people?
absolutely. I mean, this is like really exciting technology that we can change the genetic sequences or we deliver a genetic components, but at the same part of it, because it's a new type of the therapy that we haven't had much before. So a number of different safety studies needs to be conducted, and we have done a lot in the rodent system that include mouse and rat, and then going for the higher organism. In some of the cases can be dog, and in some of the cases it can be non-human primate. So those type of studies needs to be conducted before going into human.
Okay. How long do you think it's going to take to get onto the right pathways to get this done? Will it be many years, or do you expect in the next few years that we're going to have some clinical trials in humans?
Oh yes, I I definitely think that in a few years we are going to see trials in humans, and I can give like a few examples like what people have done with like gene editing technology. And one company is like Sangamo, and Sangamo has been using like order generation of the genetic feature called zinc finger nucleases. And they have done like lots of the testing, and then they are currently in phase 1 and 2 clinical trial for a few of the indications that they are looking into. So that is like kind of very first clinical trial ongoing with gene engineering, gene editing concept. And then there are a couple of other CRISPR companies, kind of CRISPR therapeutics and Editas are starting a phase 1 clinical trial with CRISPR-Cas9 and its application in 3 different areas. Like one is in the eye, the other is in blood cells.
Okay. So, any other, any big challenges that you see that you're working on that we didn't discuss in relation to this? I mean, in the mouse models, it looks like you got it. It's starting to work. What do you consider to be the big challenges ahead, the near-term ones?
Previously, kind of a number of different tries that people had, and kind of one big challenge that people faced was a translation of the results from rodent system to non-human primate, because like rodent is a great animal model system, but it's a like tiny animal and quite different to human, so that many delivery system has not succeeded after like kind of translating into non-human primate or the human. So to mitigate the risk that happens to genetics technology, we have been using a systematic approach. We have like thousands of the polymers that we kind of have in our library, and then we try to understand the biology better. And we have a big data set that we have generated in both in vitro and in vivo system. And by combining the data, we have a better prediction what is going to happen with a specific structure of the polymer nanoparticle. So those are the things that we try to overcome kind of in our next step of development.
What happens to the polymer nanoparticles, once they go into a cell? Are they digested? Are they, where do they go? Does the body see them as waste and clean them out? What happens?
Yeah. That is like very important question, because like delivery kind of we have been showing it works, but what happens to the polymer after that? We want to like kind of initially we really wanted to design a system that can be easily digested inside a cell, so that the backbone structure of our polymer, it is something that is recognized by cellular proteases, so that after the delivery, the proteases come and digest the polymer very rapidly. And that is the way to minimize the toxicity that we have with this like unique vehicle system.
Okay. Well, very good. Well, Kunwoo, what's the best way for people to find out more about the process and maybe get in contact with questions or ideas for collaboration?
Oh, yeah. We have a beautiful delivery technology, and we have many collaborators who are using our technology for various gene therapy and gene editing applications. And you can like find more information about our company at GenEdit, like www.genedit.com, our website. And also you can email like us, info@genedit.com. And that is a like kind of easy way to find more information and also like contact us.
Okay. Well, very good. Well, Kunwoo, thank you for coming on the podcast, and it's like super interesting work that you're doing.
Thank you. It's my pleasure.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.